Welcome back to the lecture series on bioenergetics of life processes. So, we have started with photosynthesis. So, today we will proceed with photosynthesis. One of the interesting thing in photosynthesis is, I have already talked to you that it consists of two photosystems, and one of the byproduct of photosynthesis is evolution of oxygen, and light is essential, as we have talked in the very first lecture of what are the prerequisite for photosynthesis to happen. One interesting aspect which was realized pretty early in photosynthesis is that there is not a linear relationship between what is the instant light falling on the leaves and what is the oxygen evolution. What does that mean? That means, say for example, I say, because it is an energetics process, so one has to keep a balance sheet to understand that how much solar energy is being used and how much oxygen is being evolved and what is the efficiency. So, it was believed that, say for example, x quanta of light or x quanta of photon is falling on a surface, then a corresponding y or x quanta of oxygen will be evolved. There should be some stoichiometry. It was observed that it is not kind of a very linear like one photon, one oxygen, not like that. It is something like after several photons the oxygen is getting evolved. In other words, you need a good number of photons to fall on the chlorophyll molecule before the oxygen is being getting evolved. That set up a very interesting idea, initial phases and eventually a hypothesis, and now kind of it is believed is that not all chlorophyll are involved in evolution of oxygen. Apparently, suppose we look at the whole structure of the leaf, there are millions of chlorophyll molecules in one cell. So, it is not that every chlorophyll will lead to the energetics reaction of evolution of oxygen. Because why we are talking about evolution of oxygen? Because that is your output. That is how you are evaluating how much, what is the efficiency of the system. It seems like there are reaction centers. In other words, let us put it in perspective. So, this is our week 4, lecture 1, and this is essentially our lecture 16. Okay. W4 L1. Okay. So, what does that mean is, say for example, we are talking about a leaf surface, okay, and light is falling, which is okay. So, we are evaluating based on the oxygen evolving out. Okay. So, it has been observed after quite a good number of photons which are hitting upon, you have one oxygen molecule which is getting evolved. In other words, the photon absorbed by many chlorophyll leads to photons absorbed by many chlorophyll leads to evolution of single molecule of oxygen. Okay. So, that brings us to a concept called reaction center in the chlorophyll molecules. That means, say for example, this is all a location of several chlorophyll molecules are sitting there, and say for example, light falls on this. So, the energy kind of get transferred. There are several forms of energy which are getting transferred, and one of them out of this will be the reaction center, from where electron will be emitted out. So, in 1932, Robert Emerson and William Arnold measured the oxygen yield of photosynthesis in chlorella. Okay. Chlorella is an easy model system to work with. Chlorella cells, which is a green algae, it is a unicellular green algae. Okay. This unicellular green algae, when exposed to light flashes lasting for few microseconds, they expected to find that the yield per flash would increase with the flash intensity until each chlorophyll molecule absorbs a photon, which would then be used for the dark reactions. But the experimental observations was entirely unexpected. What was the unexpected aspect on it? It was a saturating light flash led to the production of only one molecule of oxygen per 2500 chlorophyll molecule. Okay. So, in other words, if this is the site where there are 2500 chlorophyll molecules, which is kind of estimated based on the per unit density of the chlorophyll molecule, it was observed, and this is single flash of light. Single flash. So, one can calculate the energy what you are giving. Okay. Single flash of light. What they observed is single oxygen which is getting emitted out. So, in other words, if you draw a graph, it will come something like this. In the y axis, sorry, in the x axis you have light intensity, and in the y axis you have rate of photosynthesis. Okay. And this is the limiting value. So, this gave birth to the concept of photon absorbed by many chlorophyll, photons absorbed by many chlorophyll is funnel to a reaction center. Now, what is critical is what is the reaction center and how to identify. As of now, to the best of my knowledge, no one can really predict which one is the reaction center, and how this reaction center is being determined among all these same cast of chlorophyll molecule is also a very unresolved and a mysterious question, but it does exist. Okay. The energy level of chlorophyll at the reaction center is lower than the other chlorophyll. Okay. So, in other words, the energy level out here is lower. So, the way it works is something like this. If you kind of think it like that, these are the chlorophyll which are sitting at ground state. So, there is a light which is falling on it and which takes this chlorophyll at a higher state. There is an energy transfer to the next, to the third, and when it comes to the reaction center, so this is where the reaction center, it is sitting at a lower energy. Okay. The energy level of the chlorophyll at the reaction center is lower than the other chlorophyll, which enables the reaction center to trap the excitation. Okay. So, this is what the figure is telling you. The transfer of energy by direct electromagnetic interaction between chlorophyll and then the reaction center is very rapid occurring. So, this is the timeline or time window we are talking about, picoseconds. So, to trap such a reaction of that intensity is exceptionally challenging. Okay. So, this is all about the reaction center, what you needed to know. That kinds of, in terms of energetics, gives you an idea what is the input energy and what is the output. Input energy in terms of photons. Okay. So, this is the first point I want you to kind of take into account. The second thing is, second aspect is oxygen evolved in photosynthesis. PS is photosynthesis, comes from water. This is the second concept. So, if I put the two equations side by side, CO2 plus 2H2O, where light is falling, you have CH2O, which is your carbohydrate, oxygen plus H2O. Now, if you replace CO2 with 2H2S, hydrogen sulfide, if you remember, I talked about this, what you are going to get is CH2O, which is again the carbohydrate, plus 2 sulfur plus water. Okay. So, this is the sulfur that is formed by photosynthetic bacteria is analogous to the oxygen that is evolved in plants. So, this is what are the sulfur bacteria does. Okay. And Van Til proposed a general formula for photosynthesis, and the general formula for photosynthesis is like this: CO2 plus 2H2A, keep this in mind, this A, in the presence of light forming CH2O plus 2A, that A which is coming from that side, plus H2O. Now, what is this A? So, CO2 out here is your hydrogen acceptor. In other words, CO2 is getting reduced, hydrogen acceptor, because it is accepting hydrogen, it is getting reduced to this situation. So, this is a reduction reaction happening here, whereas H2A, this is your hydrogen donor, this one is hydrogen donor. This product, which is carbohydrate, is your reduced acceptor, and 2A is dehydrogenated donor. This is that dehydrogenated donor which has donated the hydrogen or the electron. Dehydrogenated donor. Okay. So, this is how the reaction occurs, and one can prove it very easily if you have radioisotope, like you have H218O, you have different oxygen isotope with CO2 in the presence of light, you have CH2O, and what you are getting is 18O2. So, if you see the mass of this one, which will be higher, and that is how you can figure it out that oxygen is getting evolved by splitting of water, splitting of water. So, it seems that nature learned long back how to split water, and if nature could split water and could lead to the evolution of oxygen, and of course, the other product which is getting used up here is the hydrogen. That means, for those who dream of having a world of clean energy by hydrogen evolution, nature already knows the trick, and that is the beauty of nature. It has learned in its own laboratory of billions of years by playing with its own atoms how to split any molecule, and that is the charm of studying these things. That the whole world is looking towards energetics, clean energy by hydrogen economy. You must have all seen somewhere they want to store hydrogen, that is the cleanest form of energy, and blah blah blah. What is important is nature does it. Nature does it very elegantly. Instead, just nature does not produce hydrogen in the air, it utilizes the hydrogen for all its reduction purpose. So, this brings me to that point what I was trying to highlight in the beginning, that the world or life system's energy economy is governed by a perennial electron donor. You needed a perennial electron donor all the time, because if you see out here, the world was a harsh place, lot of hydrogen sulfide, and the perennial electron donor at that time was hydrogen sulfide. All those sulfur loving bacteria and all those things survived some billions of years ago. Still, they could be seen in anaerobic conditions. But then, as the earth was cooling down, if you remember one of my previous lectures, I told you, As the earth was cooling down, hydrogen sulfide and all these things were lowering down. So nature had to search for another perennial electron source, and that's where nature picked up this wonderful molecule, water. Nature picked it up for its hydrogen supply or electron supply, but simultaneously nature, while splitting this, started evolving a very strong oxidant called oxygen. And that was that great point in the history of chemical evolution where oxygen took over. And as soon as oxygen took over, all these anaerobes were threatened. And because all iron 2 plus become Fe3 plus, everything which was there were getting oxidized. Higher oxidation states were picking up. And that's why this transition, called the Great Transition, over another millions of years, pretty much all our ecosystem started depending on oxygen. And that was the rise of aerobic situation. But still, there are places where you have the other modus operandi is still operating, where H2S is being used and is being utilized. Okay. Now coming back to another concept which I wanted to highlight in this lecture is there is something called Hill reaction, which is illuminated chloroplast evolve oxygen and reduced electron acceptors. So this is one concept. I am not getting into the detail of the reaction, how it has been done, but just you needed to know the basic message which is given. Illuminated chloroplast evolve oxygen and reduce electron acceptor. And if you realize reduce electron acceptor, that means CO2 becoming CH2O. So essentially this is that electron acceptor. If you just see the previous slide, this one, hydrogen acceptor, what is, which is essentially an electron acceptor. So you can replace that electron acceptor with another electron acceptor and prove this fact, okay? And it evolves oxygen, which is a strong oxidant. There is another interesting concept before I go into the architecture of it, is that two light reactions interact in photosynthesis. So two light reaction interacts in photosynthesis. What does that mean? Apparently, it looks like that photosynthesis is governed at two different spatial locations, a single unit of photosynthesis especially, and this is what I am talking about, the evolved plant models what we are currently more interested into. There are two specific centers. So if you go back pretty one of the very, well, let me go back and yes, out here. Now if you look into this picture, if you see it carefully, so this is where the photosystems are being placed. So out here there are two unique centers called photosystem 1 and photosystem 2. There are two unique photosystem modules which are there. So investigation of the dependence of the rate of photosynthesis on wavelength of incident light led to the discovery that chloroplast contains two different photosystems. The photosynthetic rate or the rate of oxygen evolution divided by the number of quanta absorbed gives the relative quantum efficiency of the process. Okay? So that is the energetics. For a single kind of photoreceptor, the quantum efficiency is expected to be independent of wavelength over its entire absorption band. This is not the case in photosynthesis. The quantum efficiency of photosynthesis drops sharply. So this part is very important, the quantum efficiency of photosynthesis drops sharply at wavelengths longer than, remember that, longer than 680 nanometer. Okay? Although the chlorophyll still absorbs light in the range of 680 to 700 nanometer. However, the rate of photosynthesis using long wavelength light can be enhanced by adding light of shorter wavelength such as 600 nanometer. The photosynthetic rate in the presence of both 600 and 700 nanometer light is greater than the sum of the rates when two wavelengths are given separately. This part is very important and I want it to kind of keep an attention. The photosynthetic rate in the presence of both 600 nanometer and 700 nanometer light is greater than the sum of the rates when two wavelengths are given separately. Okay. So, if you give these two wavelengths together, if you give 600 and 700 nanometer separately and add the efficiency in terms of oxygen evolution, or if you give 600 nanometer plus 700 nanometer together and oxygen evolution, you will see here the efficiency is more. Efficiency is more. So, that led, this observation is called a red drop and enhancement phenomena, led one of the scientists in this field, Emerson, to propose that photosynthesis requires the interaction of two light reactions. Both of them can be driven by light of wavelength less than 680 nanometer, but only one of them by light of longer wavelength. Okay? And the red drop graph is something like this. So, here you have the wavelength 400, 520, 600, 680, and here you have the quantum yield of photosynthesis in terms of oxygen evolved per photon, which is called quantum yield of photosynthesis. The quantum yield of photosynthesis is something very interesting to look at. If you look at the graph, it is something like this. This is that red drop. So, if you add the 700, then something like that. This drop you will not see. This quantum yield of photosynthesis drops abruptly when the excitation wavelength is greater than 680 nanometer. Okay. So, this is something very interesting which was observed, one second, which was something like this, slightly more out here, 1 second. Okay. Let me just redraw this again for you. So, it is something like this. Let's move on and something like this, red drop. Okay. So, Emerson, again I am repeating it, Emerson proposed that photosynthesis requires the interaction of two light reactions, both of them can be driven by light of wavelength less than 680 nanometer, but only one of them by light of longer wavelength. So, this is where if you use only one, then this is what is going to happen, there will be a drop. But if both of them are together, you will see a higher efficiency. So, I will close in here. In the next class, we will move on to the actual light reactions for system 1 and for system 2. Thank you.